So just to keep this panel on time, I would like right away to invite to the podium our last three panelists. And again, these are three amazing, compassionate, but also fearless clinicians, very well representing who we like to partner with at MMRF. So Irene Gabriel from the Dana-Farber Harvard Cancer Center. Irene has been specializing in a precursor condition. And then Suzanne Lange from Columbia. She's another fearless fighter who was just telling me about one of these novel trials she's going to be starting soon. And finally, Suzanne Trudel from Princess Margaret UHN, who has been pushing quite strongly in recent years molecularly targeted agents in the treatment of myeloma. So with this,
cool way to help.
There we go. All right, just to position this, again, we've discussed a lot about the immune system, but as you will hear, there's ways to intervene when these precursor myeloma cells become full-blown disease. And even when you think about myeloma, and I think Larry Boyce from Emory coined that term, the Tao of myeloma. When you think about the myeloma cells, it's a plasma cell, and there are drugs to attack the fact it's a plasma cell, but there's also a drug to attack the fact it's a cancer cell. And maybe early in the disease, the plasma cell biology of myeloma cell is an important one to go after, but as disease progresses, maybe actually you need agents that go more strongly after the cancer cell biology of these myeloma cells. Maybe the pedal is down to the metal, a bit like what Alex showed earlier for immune responses, and you almost need to throw a dynamite stick in the engine to basically stop that car from going over the bridge. So, and then there are other approaches going after macroenvironment and other clever ways to make this happen. But, and maybe just one last thing before we get to Irene. There we go. You know, a lot of the patients here in this room were telling me, so it looks like it takes such a long time to bring these new drugs. What can we do? Can we push a bit on FDA? It doesn't make sense. And we've heard quite a bit today about what we need combination of immune, all kind of agents. How do you make this happen? And this is where these approaches using these so-called master protocols become very important. As you've heard this morning from Kathy, about 50% of myeloma cells have these so-called genomic alterations that alter pathways or specific target you can go after with targeted agents. You'll hear from that from Suzanne. But the other 50%, you know, can be attacked with novel agents or immune agents. And how to make this happen faster and better, how to put these patients in the best possible bin to make this a reality. And this is what umbrella protocol, master's protocol allow us to do. And the last point here is the other way to speed up the process is maybe there are markers, and actually there are markers today that are more sensitive than what we have now, for example, with M protein, which is this top level you're seeing at the 10 to the minus 2, that would tell you ultimately years ahead of time maybe that the disease is coming back. And you don't then need to run these trials up until the M protein goes back. Maybe you use these markers as surrogate to end the trials faster or make ultimately decision for patient faster. So with this, it's my pleasure to introduce you to Irene and her beautiful comments and work in precursor conditions.
All right, well, thanks, Daniel. My name is Irene Gabriel from Dana-Farber, and I just want to echo everyone to say thank you again to the MMRF and MMRC, because you're not just disruptive for myeloma, but you're also enablers to physician scientists and to really put us together with not just foundations, but pharma companies, and truly making things happen and move forward. So I just want to thank you all again. So the whole idea of prevention of progression came a couple of years ago when we started thinking, well, as we all go for our screening programs for cancer, whether you go for a mammogram as a woman or prostate PSA or colon cancer screening, and if one of us has an early lesion, something called DCIS, ductal carcinoma in situ, or you have a colon polyp, your doctor will always tell you, well, we'll take it out early and we won't wait for you to have metastatic disease and we treat you. In fact, we never tell a woman who has an early breast cancer lesion, why don't you wait until you have bone lesions and then I treat you? Yet we do that every single day with our patients with multiple myeloma. We see patients with MGUS and smoldering disease, and every single day we tell them, you have to wait until you have lesions in your bones, until you have kidney failure, until you have fractures, and then I'll treat you. And that whole idea of watch and wait until you have symptomatic disease didn't make sense. And we started saying, well, maybe the biggest problem that we have right now, it's not that we don't have good drugs for myeloma, it's just we're waiting too long for us to have metastatic myeloma and then we treat them. And maybe by just changing the way we think of treatment of myeloma, we can make a big difference. In fact, maybe we can cure myeloma by early therapeutic intervention. So 2 years ago, we started at Dana-Farber something called the Center for Prevention of Progression. And it wouldn't happen unless so many people really came together. It was really a vision from, of course, our leadership, but it was also a vision from many people. Adriana, who's here in the group, who's truly a business leader, again, coming back to ideas of using science, but also bringing in the business development to really put it together. But also it's combinations of working with pharma, with foundations, and many other people. And the whole idea came along with us saying, let's define who are those patients with early precursor conditions, MGUS and smoldering. Let's define who will truly progress to real myeloma early on. So whether you do mutation sequencing of their tumor cells, of the blood biopsies, so we can look at all the serum samples at different time points and look at circulating free DNA or circulating tumor cells, but also looking at the immune microenvironment early on, because we know that that's early on can prevent progression if you can harness the immune system and not wait for it to be so disruptive later on in the disease. And the first thing we said, then let's really get the science up and running, and we collaborated with the Broad Institute on doing that, which is the biology part of this. Then we came to a point of saying, well, if there are data banks and large data sets from many people before, let's go back to the retrospective data and not have to wait 10 years and 20 years to get our data. So we're collaborating with Mayo Clinic that has a huge data bank of 50 years of follow-up, thanks to Dr. Kyle, who's an amazing physician who thought of collecting samples 50 years ago. And we're trying to get all of those samples to look at them and sequence them and have the answer today and not 10 years from now. And then the third part of this was the P crowd. And truly, this was an idea that came of really empowering our patients. And instead of doing crowdsourcing for money, we're crowdsourcing for patients. And the idea came along that we don't want to wait until patients come to Dana-Farber and then we get your sample or your tumor cells. And we don't want to wait for academic centers to collaborate with us and give us the samples, because that's too long and it takes too much effort. And we said, let's just go to the patients directly and tell them, here's a website, log in, put your information on it, send us your samples, and we'll collect the data. And we came to a P-Crowd initiative. So this is online. Anyone can sign up to it. The consent is online. They complete a very simple survey. It gives us access to their data records. We're hoping that this will now become even electronic so that we can get all the electronic medical records put together. We're working with the Broad Institute to integrate all the information between clinical data, genomic data, immune therapy, as well as clinical trials. We're tracking the health. We're now collaborating with patients like me to track the health of the patients, to look at quality of life, as well as survey their symptoms and signs. And then, of course, we're collecting large numbers of samples, and then we're working with many people, not just the Broad, not just MMRF, but also many foundations and many companies to really enable this to happen so that we truly understand who is the patient who will progress and who's the patient who will not progress in their lifetime. And once we do that, and because we have this urgency of let's really make a difference fast and not wait for us to understand all of this, we know already that many patients with high-risk smoldering disease and even high-risk MGUS and low-risk smoldering may already be potentially ready for clinical trials. So we started designing clinical trials that harness, again, the immune system, whether it's vaccine therapy, whether it's the immunotherapy that you just heard about, elotuzumab, daratumumab, and many others, as well as nivolumab, so that we can put them together to try and understand already, can we prevent progression? Can we make a difference in those patients? Could we potentially cure some patients early on? And then finally, after we do all of this, we're starting to say, well, if we can do this, if we can understand who progresses and who doesn't, and if we can understand molecularly how can we target those patients and potentially make a difference in clinical trials, can we screen patients? The ultimate goal is that when you go for your checking your cholesterol level and you're checking your heart rate and you're checking your colonoscopy, why don't you just get one blood sample and check if you have M-spike or not? Because if we have familial history, if we have African Americans who have a much higher risk of developing myeloma, and if we have other risk factors, just by knowing that you have an M-spike and you may progress to myeloma, if you can understand this and prevent it early, maybe myeloma will be actually a preventable disease, and we don't need to treat patients later on because we'll just prevent the disease from happening. So with that, I'll stop.
Thank you so much. So with this, Suzanne?
I mean, thank you so much. That was kind of, very nice and interesting, and I think that's very important work. I come from a slight different angle. This morning, I'm coming straight from clinic. I saw around 15 patients. I rushed a little bit. I apologize to my patients that I did this, but I had a good excuse to come here. I hope you hear me. And a couple of times I had a dilemma. I had a dilemma with my patients and what to do. So I started one patient on Ninlaro, Revlimid, and Dex, and the next patient I started on Ninlaro, Dex, and there was another patient I started on daratumumab, pomalidomide, and the patients asked me, Doctor, is it the right choice? Why are you doing this? Is it the best for me? And there was a big dilemma. I tried to explain my decision tree that I said, okay, you have a high-risk multiple myeloma, hope we can identify high-risk multiple myeloma, or you prefer to be on an oral drug, or you come from upstate New York. But I didn't really have a good answer, to be very honest. I couldn't tell the patients, you have this multiple myeloma, that's why you need this specific medicine. And I would slightly change my title. We have a dilemma, but we don't have the dilemma of too many drugs. That's impossible in multiple myeloma. But I think the dilemma is how can we fine-tune the current drugs, and how can we use our knowledge and the tools in order to give the best treatment to our patients. And we had, I think that's, sorry, I'm playing around with this one. Just last year we had 5 new approvals, and I'm sure you heard a lot about that. Panabinostat is an HDAC inhibitor. We have a new combination of carfilzomib, lenalidomide in combination as an approved drug. We have daratumumab as an antibody, ixazomib, a new monoclonal, new proteasome inhibitor, and elotuzumab. So you see, in addition to the options we have right now, there are even more combinations. And what you can see here on this slide, and I brought that up in order to also show you the dilemma we have as doctors and the difficulties. You see that this additional 5 approvals really result in many more options. So you can combine your antibodies with either an IMiD or with a proteasome inhibitor, or you can combine in clinical trials your PD-1 inhibitor, pembrolizumab or nivolumab with an IMiD or without an IMiD. So there are a lot of options right now which really bring us into a situation in which we say, what is the best for my patient? And in addition, there's research ongoing. And what I mentioned here is selinexor, that is a new medication. It's a first-in-class exportin-1 inhibitor, an interesting drug, because what it is doing is usually you have proteins, tumor suppressor proteins in your nucleus, and with a protein, with the XPO1 protein, those tumor suppressor proteins are shuttled out into the cytosol. And what that exportin-1 inhibitor is doing, it inhibits the transport of the tumor suppressor gene, for instance, of tumor suppressor proteins from the nucleus into the cytosol, and increases tumor suppressor protein presence in the nucleus and brings the cell back to a normal regulation or can induce apoptosis in case there are abnormal mutations, as Ralph Kleins mentioned already. So those are interesting drugs, and I picked that because we expect at the ASH meeting this year's the presentation of ongoing trials, selinexor in combination with proteasome inhibitor, but also in IMiDs, so very promising drugs. And more drugs are really coming and are on their way. So that's why for us, I think it is really difficult to, or we really need to know what is the best sequence of the currently approved drugs. What are the best combination of the approved drugs? So should I first start with an IMiD and a proteasome inhibitor, or should I go, for instance, should I combine that with a monoclonal antibody? We also need to identify patients who benefit most from specific combinations. What we do right now, very often patients ask me, what are the side effects? Will I have the side effects? The problem is I cannot really tell patients, you will have that side effects. I can only say, based on the current data we have, 30% of the patients develop neutropenia. And I think the COMPASS study is really important here to help us, not only to pinpoint certain mutations, but also go back and say certain drugs work better in patients with bone lesions, for instance, or other patients have more side effects. So I think with the help of this study, we will identify a patient population responding to certain combinations. It will help us to better identify high-risk patients. I have to say I still really struggle with the identification of high-risk patients. I don't think that we are really able to say who is a patient who has a high risk. And last but not least, and I think the other Suzanne will talk about that, it will enable us really to target certain mutations. We heard a lot about mutations, and I think the problem what we have is you can have multiple mutations. One can lead to the other, which is a driver mutation, which is a really critical mutation that results in an aggressive cancer. And having said this, I would like to give that to Suzanne.
To the other Suzanne. I think it speaks to our generation, Suzanne's, lots of them. Okay, my slide next. Okay. I would like to echo Irene's comments about how the MMRF has really been enabling, and my interest has long been in the area of precision medicine, and I'm happy to say that precision medicine is ready for prime time in myeloma, and a lot of this has been due to the great efforts of the MMRF to get us to this point. So I think really the MMRF has really been driving the introducing next generation treatments for patients with myeloma, and this is building on the cataloging of genetic abnormalities that have been identified in large genomic efforts such as COMPASS that you've heard about today. So we're identifying mutations such as genes such as RAS, and you'll hear a little bit about FGF receptor 3 today. That can be exploited therapeutically by using drugs that can selectively turn off these genes that are acting abnormally in the cancer cells and are really hopefully Achilles heels for the cancer. These we call the bullets. So I think if you've heard already a lot about today is that myeloma is not one cancer. And this is probably best illustrated in this figure here. It looks a little bit complicated, but this is a gene expression profiling of RNA derived from tumors from over 300 myeloma patients. And each column of red and yellow dots represents a sample from one patient. And you can see in this picture that you grossly see 3 patterns appear. And this represents 7 subsets of myeloma patients. And highlighted here is one of these subsets of patients that represents about 15% of myeloma patients. And these patients are characterized by having a translocation of chromosome 4 and chromosome 14 that can be detected in the clinical lab by FISH analysis, which is often done in patients at diagnosis. And the significance of having this 414 translocation is that tumors that have that translocation express a gene called FGF receptor 3 that has the potential to be therapeutically targeted with drugs that can turn off the function of FGF receptor 3. And with the support many years ago of the Multiple Myeloma Research Foundation, we actually showed that if you turned on FGF receptor 3 in cells so that it was constantly functioning on all the time, that it generated tumors in mice. And this was similar to what we saw with another very well-known driver, cancer-driving mutation of RAS. And if we treated these mice with a drug that turned off the signaling of FGF receptor 3, we could see that the tumors no longer occurred in the mice. So the next step, once you've sort of found this mutation and you think that the gene that is mutated or abnormally functioning in a cancer cell, and you've shown that it is potentially an Achilles heel, that the cells require that mutation for survival, is then now to test drugs towards this particular target in the clinic. And this has proven successful in many cancers, BRAF mutated melanoma, EGFR mutated lung cancer, as well as in colon cancer, and another disease very familiar with myeloma, Waldenström's macroglobulinemia. And recently we've also had the proof of concept that this type of approach will work in myeloma, when during the first sequencing effort by the Multiple Myeloma Research Foundation, the MGI initiative, we identified that there were some patients, small percentage of patients had mutations in BRAF. And there was already a drug in the clinic to treat melanoma, because most melanoma patients have a similar mutation. And what was shown is those patients who were treated with these BRAF inhibitors, you saw a decrease in their M spike and a shrinkage of their plasmacytoma. So this was really a proof of concept that this type of precision medicine approach would work for myeloma. And the great thing about the COMPASS study now is that we now know that not only BRAF, but approximately 50 to 60% of myeloma patients probably have mutations that are potentially actionable, that we can potentially target. And also we know that many of these targets, there are already drugs out there in the clinic that can be used to treat patients with these mutations. Now, the dilemma remains that obviously we have to be able to screen patients for the mutation. This requires that patients have bone marrows, which I'm sure many of you in this room know is not the most pleasant thing to go through. And so we have been working over the last 2 years to develop an assay where we hopefully will no longer need bone marrows. We call it the Milestone Project, Myeloma Stop the Needle. And essentially what we know is that tumor cells actually release into the circulation fragments of DNA. So if we can collect the DNA from the plasma in blood, a small fraction of that DNA will be from the tumors. And now with very advanced sequencing techniques, we can actually sequence those small fragments of DNA in the blood. And we actually showed, when we looked at 5 genes, 3 of them were genes that were highly mutated in myeloma, RAS and RAF, that we were able to detect in 97% of cases the mutations from the blood tests that matched the bone marrow test. And so now we're expanding this panel to a more extensive list of genes that we think are important for myeloma, many of them which we think are potentially targets that we can use therapeutic avenues for patients with myeloma. And the plan is we've gotten funding from the MMRF to fund that initiative. And we also have funding from our own institution to move that platform from the research lab to the clinical lab so we can offer it to patients. And the other important finding there is that we hope that we will be able to use this platform to then track patients over time that are going on these precision medicine trials without having to do repeated marrows. So just to summarize, as I think we've heard a lot today, is that myeloma is not one disease, and that DNA sequencing of myeloma patients' tumors are providing researchers a comprehensive catalogs of key genomic changes, and this has really been accelerated by the COMPASS study. And these hold promise for precision strategies to treat myeloma. And we are now in prime time ready to go with a number of precision medicine trials. One of them we will be conducting at our site, which I'm very excited about because it is a culmination of many years of work where we will be testing a drug from Janssen. And I have to thank the MMRF for really helping us get this trial up and running and initiating that collaboration with Janssen to get this study up and going that will be specifically for the 414 patients. But in addition, there are other trials ongoing through the MMRC, including studies of inhibitors of the RAS signaling pathway, also a study specifically targeted to patients with 17p deleted myeloma in combination with ixazomib and dexamethasone, which is being led by the Mayo Group. And then you've heard about the master protocol, which is under development, work that is being done in collaboration with the NIH, which we're all very excited about. And as I mentioned, also important challenge is now matching the therapy to the patient, and hopefully we will soon have less invasive ways of being able to do that for patients. Thank you for your attention.
Thanks so much, Suzanne. So we have maybe about 15 minutes left. Let's go first to a question that I have for the group here, and maybe we can go to a few of these questions from the audience. And I think let's start with you, Suzanne, but that's for the entire group. As I said, how do we test all these hypotheses coming out of Compass and others about these potential new combinations? How do we test that in a way that we find a signal or not rapidly, and then move these drugs in an expedited fashion toward the FDA approval? We discussed master protocol, but I'd like to hear your thoughts on that, or other approaches through which, as we've always done, and we meet regularly with FDA and other groups, how to accelerate testing all these various combinations we've heard today from the various groups in a way that will benefit all of our myeloma patients. But let's start with you, Suzanne.
Yeah. I think we made enormous progress in the response, achieving good response rates in multiple myeloma, especially in newly diagnosed patients. I think we all agreed that newly diagnosed patients can achieve response rate up to 90%. So I think the problem starts more and more when we go into the relapsed refractory situation, when patients received, the second, the third line treatment, and it comes to a decision where you kind of, had an IMiD and you had a proteasome inhibitor and you have multiple translocations, which is the best treatment, which is a treatment that has less side effects. I think, and that's what I see right now, that the studies like the Compass studies, large kind of studies who really collect data and can retrospectively combine or say give us information on the response rates, on the outcome, on the overall survival, on the sensitivity of the disease. I think that those large trials are really needed right now. I really don't foresee another way to do that. The doctors, and I'm very honest, we are also overwhelmed with the amount of data and information when we start to treat our patient. It's a gene expression profile, it's a Columbia Cancer Panel, our mutational analysis. It's the FISH, or does it play a role? It's the karyotype. It's the patient itself. It's the albumin. It's the beta-2. I mean, there's so much information for us that at the end, we treat most of the patient the same in the beginning. We rely on data and we say, based on what I have, there's a 90% chance of response. There's a 30% chance that you might have, neutropenia. And in this situation, we are pretty much okay and feel comfortable. But again, the third, fourth line treatment, relapse, what can I do? What is a mutation I can target right now? And right now, the only way to do that is large trials. That means not 20 patients, that's hundreds of patients, thousands, if we could have kind of, the data. The problem is that, and I know that there's industry here, that very often industry does not really have the interest to do those trials. I mean, the primary interest is to produce a good drug that helps a patient, but when we have 10 different medications, what is the best one? And I think the Compass trial is kind of, one of the most kind of, helpful trials in order to pinpoint, this combination really worked in this patient group. It will take a while. That's nothing we can achieve, in a year. That takes maybe a decade to get those data, but we have to start to do that. And in addition, I have to say, those data also help research, future research, because what researchers can do, for instance, if you find the mutations, I'm doing this, I find an interesting target, I go back and say, does those patients have, for instance, bone lesions? So it's not only for the treatment right now, it's also for research. But again, right now, I think is collecting data and see who benefits most from which treatment.
Okay, yeah. So it's a few things that to go back to a few things you said. I think one of them is, I agree with you, we need these large hundreds of patient master protocol type approaches. And this is where foundations like ours can actually sit down with our pharma partners and, as some of sometimes help to convince our pharma partners that we believe that a given arm or a given trial should happen. And thanks to everyone here in this room, we also have to put some of our own money into it, but we believe in this. Suzanne, Suzanne Trudel, how about you now? Is there a more precise, a more clever, a faster way to make these things happen?
Well, I think we have to use both. I think I totally agree with Suzanne that there's a lot of power in numbers, and the more data you collect, and you can start to look for patterns like we did with the gene expression profiling, for instance. But I think also to get things done very quickly, there has to be other ways that we can also try to expedite new treatments, especially sort of things, precision medicine approaches, or just trying to find combinations that may be more effective. And I think there's 2 studies that are sort of actively being in development in the MMRC, which I think are really addressing that. And one is the master protocol, where, the only way that we can do these precision medicine trials is to do a broad number of targets, because obviously you want to offer all patients, some potential therapeutic avenue, but also you need a lot of sites involved, which we have that power through the MMRC, with a lot of sites being involved and a lot of collaborative groups that we can initiate studies with, so that we can test in small numbers if it's these sort of precision approaches, and see whether there's a signal, and then if there is, then look at doing larger data set, but just kind of a proof of concept in small numbers of patients. And then there's the I-SPY study, which is essentially looking at adding on to a backbone and seeing if we have 10 patients, we add another drug, a novel drug, are the responses better? And in our case, are the responses deeper, because we already have a very high response rate now? And then go on and use that same backbone, add another drug, and then another drug, and see what in small numbers of patients gives you the most effective readout, and then start to expand into a larger patient population. I think that will give us a very quick answer that can then sort of be further explored.
So Irene, I think through your presentation you gave a great example of how going directly to patient, we can go beyond these 1,000 COMPASS patients. We can go to the 5,000, 10,000, all patients, the million-plus patients with precursor conditions to myeloma. How do we make this happen in this arena of precursor disease?
Yeah, so I think that's the new power of, and Kathy brought it up today, is like we have an amazing power now. Patients have the power, and we should just empower them and let them take it over and not be limited by what we do right now. So thinking a little bit beyond what we usually do. So I absolutely agree with Suzanne, but there is that fierce urgency of now. We have to do it now, and we have to stop thinking of the very traditional way of doing designs of studies. I think empowering patients to put all their samples and sequencing it at a larger way so that we can truly get the small numbers of patients who have specific mutations that we can target in a better way. Encouraging patients to truly go on clinical trials. We have so many patients in the US, yet the numbers of people who go on trials is minimal. Compared to Europe, where, again, because probably of access of clinical trials, of access of drugs, it's different. But we can do something to make a difference, and we can do it now. And I think I absolutely agree, yes, there is the baseline, drugs that we use for plasma cell biology. But Kathy brought it up today in the morning. We all have different genomes, and yet we're all being treated with the same 3 drugs. they're very good 3 drugs, but we need to go beyond that and trying to understand what is my genome before therapy, what's my genome after, did I have a specific subclone that would be leading to drug resistance later on. So that adaptive design of working truly between the clinician, between the patient, and truly quickly with precision medicine to try and understand what happened before and after so I can change therapy is the way to go. I think MMRF can absolutely do that. And I don't think we are at the era of doing that. It is the era of amazing science that we can just use it and harness it for ourselves.
I told you they were fearless.
I think also following up on that, I think really important is the correlative studies within those trials to really understand how the drugs are working, because still we don't understand how a lot of myeloma drugs work. And what are the mechanisms that are leading to resistance? So I agree with you, more trials, but also more correlatives in patients donating their marrows during the process of the trial to really understand how things work.
Yeah, more trial and more intelligent trials,
Exactly.
which is what this consortium prides itself into. Let's go into a couple questions. Here's one which I believe I know the answer, but I'll put it out to you guys. If the FDA approves a new drug, how long does it take to be listed on the NCCN guidelines? Well, there is a process. One of you want to dare to explain the NCCN process?
Who's a member?
Ken is a member, so I'm not talking.
No, I think there needs to be a certain evidence that the drug is potent and it's working, and there needs a publication in order to really justify. There are different categories of evidence before the drug can be listed, on the NCCN guidelines.
Yes, and I don't think there is a time to it. It's more of a process. There are regular meetings where the top investigator meets with the regulators and decide which of these should make it or not to the NCCN guidelines. But again, we've been, as a foundation, present at some of these meetings. This is, again, where we can play a very important role with the regulators. Yeah, I think that's pretty much it for these questions. All right, so any closing thoughts from this panel? Let's start from, I guess, right to left. Irene?
Well, I would close by saying you are doing an amazing job and keep doing it. I think, we had just said that you're truly enabling by putting all of us together, by putting physicians and scientists and pharma and foundation and everyone together. But not only that, but truly thinking outside of what is the traditional way of thinking and bringing the business model into science. And I think that's the way to go. Big data is a huge power for us. And really using it for advancing science will make a big difference. And I'd love to see the next 10 and 20 years of our life really changing myeloma and curing it at an early precursor condition.
Thank you, Irene. Suzanne?
Being the oldest among
We're not
us,
talking about age.
I have to
not
say,
talk about
no,
age.
I have to say, it's really great to see the progress on multiple myeloma. As I started in Ken Anderson's lab as a postdoc in 1999, basically I remember that Tiro Hideshima tested Revlimid, which had a name CC5013 in mice, and it was a blood paper. We were all excited, and subsequently, a couple years later, the first study, and seeing really the number of new drugs we have. I mean, it's amazing, and I think we are all blessed to have so many drugs right now available. It's unbelievable. The monoclonal antibodies absolutely changed the game. I just had a patient. I also used daratumumab for amyloidosis. One cycle, all the plasma cells were gone. It's amazing. So to see that really kind of, it gives me a huge pleasure. And what I wish is, and I think as a doctor, I owe that the patients to have a better fine tuning, to go into a patient and say, you are 75 years old, you want quality of life, you want to spend time with your children. Can I recommend an oral treatment, or should I push you into carfilzomib twice a week? Those are questions each day, and they bother a doctor, and they are most important for the patients to use their time wisely. All our time is limited. And having said this, what I really wish, and that's why I admire really Cassie for kind of her enthusiasm to bring people together. What we do in the States, we are really great in research. We are really great in bringing drugs to FDA approval, but we are really bad in finding out and working together what is the best for our patients. So having Europe as a role model to do large trials and do them easy and not that expensive, that would be my wish. So what we know right now, we work on the fine-tuning, but we should take a deep breath and see, how can we combine the current drugs best and what is the best for our patients in which sequence.
Thank you. Suzanne?
Wow, I think pretty much everything has been said, but really echoing the fact that we have a lot of tools in the toolbox now. And I'm happy to say that we've been doing some early phase studies at our site with some totally new drugs that haven't been talked about here, and we're seeing activity with those. So exciting, there's more stuff coming. But agreeing with Suzanne that we have to learn how to use them, which combinations, what sequencing. We have to sort all that out. I think we're well on our way with the COMPASS data and these databases to try to figure some of those questions out. And also being very intelligent. The MMRC and the MMRF have been so successful in raising so much money and have gotten us so far. But we have to be intelligent in how that money gets spent. And I think we've brought all the right people to do that. And I think we're going to keep seeing more and more improvements over the next 5-10 years. I mean, we're actually starting to talk cures, which I wouldn't have said 5 years ago.
Thank you, Suzanne. So I'd like to thank all of three. Thank you so much again for being on this very interesting panel. And with this, let's give it to Anne.
I really just wanted to say a couple of things. One, Suzanne Lynch, I really applaud you for being honest to say, these patients come and I'm not sure what to do, because those are the questions patients have. And I think a lot of patients think their doctor knows, oh, my doctor will know what to put me on, but the data doesn't exist. So honestly, thank you for being honest. And, it will take efforts like master protocols. It will take efforts like direct to patient, what Irene's done, what you heard about from Kathy, to collect this real world data to answer these questions, not only for patients, but for the doctors that treat them. And then, as Donna pointed out earlier today, what exists in breast cancer needs to come to myeloma, the standards, the pathways, so that patients understand, I have this type of myeloma, here's the right sequence, here's the right treatment for me, here's the right sequence. And again, as a third party, as someone you've heard from many of our partners say, that we bring everyone together, it's really our obligation to make that done. And then the final thing, because Suzanne went very quickly through the drug selinexor, I just wanted to highlight that for any of the patients in the room who may have been through a lot of the therapies. That drug right now is showing success in patients who have failed both IMiDs and both proteasome inhibitors. So Revlimid, Pomalyst, Kyprolis and Velcade, and also some pentarefractory, a new term I just heard last week, which is daratumumab as well. So I just want you to know that if it wasn't clear what's coming next, that is a really exciting new drug that is moving quickly forward.